Okay, so now we look at the process of DNA replication. Okay, so let's say this is the double-stranded of DNA. So firstly, for process of DNA replication, it will start with DNA helicase. So this DNA helicase will bind to the double-stranded of DNA and it will unwind and separate the double helix strand of DNA. Okay, so first kali awak mesti kena tahu, since DNA is double-stranded, so means that dia ada first parental DNA, okay, its strand is from 5' to 3', and then the second strand is 3' to 5'. Okay, so once DNA is already separated, so each of this strand will act as a template to synthesize a new DNA. Okay, because of what, awak mesti kena tahu lepas ni the new strand of DNA, satunya kita panggil sebagai leading strand, satu lagi kita akan panggil sebagai lagging strand. Okay, and then next, once DNA is already separated, so single strand binding protein will bind to each of the DNA strand and then it will stabilize the structure of DNA. Stabilize the structure of DNA means that it will prevent repairing of DNA double-stranded until the strand is already copied. Okay, another one adalah sebenarnya Topoisomerase ataupun DNA gyrase. Okay, so actually DNA gyrase will prevent excessive coiling of the double-stranded DNA. Okay, so once DNA is already separated, okay, awak akan nampak dekat sini, this one kita panggil sebagai replication fork. So replication fork represent the Y structure of the DNA once it is already separated. Okay, so kita tengok dulu one by one. Okay, this strand, okay, kita tengok dulu untuk parental strand which yang run daripada 5' to 3'. Okay, so next enzyme is primase. So what is the function of primase? Actually, it will synthesize RNA primer. So let's say ni adalah RNA primer. So awak mesti kena ingat the direction of the DNA replication must run from 5' to 3' untuk new strand. Okay, so RNA primer actually it will consist of RNA nucleotide to initiate the process of DNA replication. So once RNA primer is already synthesized, okay, next will come DNA polymerase 3. So the function of the enzyme DNA polymerase 3, actually it will add free nucleotide which is complementary to the bases of the template. Okay, so let's say the template strand, okay, the bases C, T, A, G, C. So complementary means C must be pairing with G, T must be pairing with A must be pairing with T, G will be pairing with C, and C will be pairing with G. So all of this, actually this is the new strand of DNA. So the new strand of the DNA must be complement to the template. Okay, so DNA polymerase 3 will add DNA nucleotide continuously towards replication fork. Okay, so dia akan tambahkan DNA nucleotide secara berterusan towards the replication fork. So, since the new strand synthesize continuously towards replication fork, so this strand we call as leading strand. Okay? Kenapa kita katakan dia synthesize continuously? Since the parent strand ni adalah 3', so the new strand dia mesti kena anti-parallel dengan dia punya template. So since template dia ada 3' dekat sini, so new strand must be 5'. So if the DNA strand is synthesized continuously towards replication fork, so this one kita panggil sebagai leading strand. Okay, so next, what is the difference with lagging strand? Okay, so remember the direction of DNA replication must be from 5' to 3'. Okay, so apa perbezaan antara lagging dengan leading? Kalau leading strand, it needs only one primase to synthesize RNA primer. Tapi kalau awak tengok beza dekat sini, it needs more than one primase to synthesize RNA primer. Because of what? Since dia adalah lagging strand, so dia punya direction dia adalah the direction of DNA replication also same from 5' to 3', but it runs away from the replication fork, or the DNA is synthesized discontinuously from replication fork. Kenapa discontinuously? Awak kena tengok dekat template dekat sini. Template dia adalah 3' to 5' here. So, ia akan complement dengan the template. Okay, maksudnya new strand dekat atas ni mesti kena 5'. Okay, yang bawah ni mesti kena 3'. Okay, so since the DNA synthesize 5' to 3' away from replication fork. So means that this strand kita panggil sebagai lagging strand. Okay. So the concept is same, means that primase will synthesize the RNA primer, and then DNA polymerase 3 will add DNA nucleotide which is complement to the template. Okay, but for lagging strand it needs more than one RNA primer. Okay, so means that kamu akan nampak di sini akan terbentuknya short fragment of DNA. So this short fragment we call as Okazaki fragment. Okay, and then next enzyme is DNA Polymerase One. So the function of DNA Polymerase One, actually it will replace RNA primer with DNA nucleotide. Okay, and then lastly, awak akan nampak dekat sini adanya enzim DNA ligase. Okay, so what happened to the short fragment of DNA, or we call that as Okazaki fragment, it will join by DNA ligase. Okay, so dekat sini awak mesti kena ingat apa perbezaan antara leading strand and also lagging strand. So actually both leading strand and also lagging strand, the strand of DNA will synthesize in the direction of 5' to 3'. But the difference for leading strand, the direction of DNA replication or the new strand of DNA is synthesized towards replication fork, while for lagging strand it was synthesized away from replication fork. Okay, and then another difference you can give, for leading strand it needs only one primase to synthesize RNA primer, but for lagging strand it have more than one primase to synthesize RNA primer. Okay, so that's all. Thank you.